Hello, my name is Skylar Davidson, and I'm a third-year student at the UNLV School of Medicine, also known as the Kirk-Kerkorian School of Medicine. My research focuses on the field of neurology and asks the question, does the presence of type 2 diabetes affect cognitive decline in Alzheimer's disease dementia? The other authors and organizations associated with the research are listed as well. Moving on to the introduction, there are more than 6 million individuals diagnosed with Alzheimer's disease in the United States, 72% of which are age 75 or older. Alzheimer's disease is best known as a chronic form of dementia with symptoms such as deficits in short and long-term memory and loss of executive functions, which eventually lead to premature death when compared to healthy aging. Type 2 diabetes mellitus is defined as an insulin resistance in the body and is estimated to be the predominant diagnosis of 90 to 95% of the overall 34 million diabetics in the United States alone. Even with proper glycemic control, there are many complications associated with type 2 diabetes that include cardiovascular disease, chronic kidney disease, nerve damage, and hearing and vision loss, to name a few. Furthermore, cognitive impairment has been shown to be a possible complication for individuals with type 2 diabetes as well. One such example of the cognitive decline observed in type 2 diabetics is from a 2016 study where executive function tasks were compared among a demographically matched healthy control group, type 2 diabetic group, and Alzheimer's group. The study showed that performance of the type 2 diabetic group in verbal and visuospatial tasks was worse than the control group, although better than the Alzheimer's group. There are a variety of proposed mechanisms that attempt to show the underlying relationship between Alzheimer's and type 2 diabetes, but there is currently no accepted mechanism. Factors such as insulin resistance, advanced glycation end products, inflammatory responses, and oxidative stressors are all being studied. The late-stage pathophysiology of Alzheimer's is even shown insulin resistance in the brain similar to that of type 2 diabetes, enough so that Alzheimer's disease has been referred to in prior studies as type 3 diabetes. While past studies have focused on the development of mild cognitive impairment to Alzheimer's disease in type 2 diabetics, the specific aim of our study was to determine whether a comorbidity of type 2 diabetes in Alzheimer's patients is associated with a difference in the rate of progression of cognitive decline compared to Alzheimer's without type 2 diabetes. In order to analyze a large data set with homogeneous entries, we decided to use the National Alzheimer's Coordinating Center, also known as NAC. Their database receives longitudinal data from all Alzheimer's research disease centers spread across the United States. The uniform data set was established by them and began the use of prospective data with clinical evaluations collected annually on the subjects. The Uniform Dataset version 3 was the source of our data for the present study, as it implemented data as recent as of March of 2015 and includes diagnoses of diabetic types and visits. We focused our analysis on the comparison of two specific groups: Alzheimer's patients with type 2 diabetes and Alzheimer's patients without type 2 diabetes or any other type of diabetes. Our search criteria narrowed down the uniform dataset to 3,055 participants in total, with data obtained from 2015 onward and with a primary etiological diagnosis or cause of cognitive impairment of Alzheimer's disease. The AD with type 2 diabetes group consisted of 359 patients, and the AD-only group contained 2,696 patients. Demographic variables were obtained for both groups. These variables include sex, ethnicity, race, marital status, the taking of diabetic medications, and other variables. The Neuropsychological Battery is a form that is completed by ADRC staff to assess cognitive functions in patients. Selected tests from this form were used to provide longitudinal data to determine the differences in Alzheimer's symptoms between the two groups of our study. 16 cognitive function tests were selected that looked at language, cognitive executive function, and verbal and nonverbal memory for each patient. The clinician dementia rating, also known as the CDR, from the uniform dataset also provided a subjective measurement in subtle declines in cognitive function and at what age cognitive decline began according to the clinician present at the time of the first visit. Statistical analyses were carried out via SPSS software version 26. A spaghetti plot was used to visualize the progression of the neuropsychological battery variables with the mean values calculated for each test score and then plotted over a timeline by visit number. Levine's test for equality of variance was used to correct for the size differences between the two groups in the study. The demographic characteristics of both groups are shown in the table on the slide. The Alzheimer's with type 2 diabetes group had a larger male population at 52.1%, while compared to the Alzheimer's only group had a higher female population of 54.1%. Additionally, the Hispanic Latino percentage was more than double in the Alzheimer's with type 2 diabetes group at 14.8% compared to the 6.8% in the Alzheimer's only group. Interestingly, according to the American Diabetes Association, the rate of diagnosed diabetes is around 12.5% in Hispanics as of 2018, which matches with our dataset. Additionally, the Black, African American, American Indian, Alaskan Native, and Asian population proportions were also significantly more than double in the AD with type 2 diabetes group compared to the AD only group. There were also significant differences with p-values less than 0.001 when comparing the Alzheimer's with type 2 diabetes group to the Alzheimer's only group in terms of current use of diabetic medications, with 67.4% of the Alzheimer's with type 2 diabetes group reporting current use of diabetic medications, while 98.6% of the AD only group reported no current use of diabetic medications. The Alzheimer's with type 2 diabetes group had a mean beginning age of cognitive decline per clinician's assessment of 72.04 years, with standard deviations of 8.47 years compared to the Alzheimer's only group with mean ages of 69.15 years with a standard deviation of 10.33 years. Moving on from the demographic variables, the analysis of 16 different cognitive function tests selected from the Neuropsychological battery form showed no significant difference in baseline scores and follow-up scores at each visit between the type 2 diabetic and non-diabetic groups. The figure shown in the slide shows the mini mental status exam and the Montreal cognitive assessment scores for both groups. Both of these tests function as a gold standard to evaluate the overall cognitive performance of patients with dementia, and we observed that over the course of 5 visits of 5 years, the test scores did trend toward a decline, but the type 2 diabetic status did not seem to affect the overall scores. It can be seen in the figure that the MoCA scores at the individual visits 1, 2, 4, and 5 did show a significant difference between the type 2 diabetic and non-diabetic groups overall, but there was no significant clinical difference in the MoCA scores between the two groups over a 5-year period with a p-value of greater than 0.05. Similarly, with MMSE scores, there were no significant clinical differences noted over 5 years, with a score change from 21.76 to 20 in the AD with type 2 diabetes group versus a 22.22 to 19 point decline in the AD only group. The figures shown in the next slide summarize the tests that examined the language function of participants. Figure 3A shows the results of the category fluency test, specifically for animals and vegetables. The goal of this test is to name as many animals or vegetables as possible in 60 seconds. The animal naming test showed a change from 12.1 to 10.57 and 11.94 to 10 in the AD with type 2 diabetes and AD only groups respectively. Similar results were also observed in the vegetable naming test. Moving on to figure 3C shows the results of the phonemic tests and the multilingual naming test, also known as MINT. These tests essentially keep track of the number and correct amount of F and L words said in 1 minute. It can be seen from the graph that there was no significant difference in either of these tests as well. Figures 4 of the study reported the selected trail making test and number span test forward, which examined frontal executive functioning. The trail making test is depicted specifically in figure 4A and involves 2 distinct parts. Part A of the test has the participant connecting dots that are increasing in numerical order, and the total number of seconds to complete the task is then recorded. Part B of the test also has the participant connecting dots, but there are numerical and alphabetical dots present, and the participant must go in order for both while alternating between numerical and alphabetical values. These tests focus on gauging the participant's attention span. Figure 4A shows that there was no overall difference in terms of score change for all tests related to frontal executive function, both for individual visits and over a 5-year period with p-values greater than 0.05. Tests of verbal and nonverbal memory were done with the Craft Story 21 recall and the Benson complex figure copy in Figure 5. Figure 5A specifically shows the results of the Craft Story 21 recall, both immediate and delayed. where the participant was scored based off the total story units recalled verbatim. Visits 1 through 4 in both the immediate and delayed Craft Story 21 recall tests show significant statistical differences for each individual visit between the two groups, but clinical differences were not significant over 5 visits, with p-values greater than 0.05. In conclusion, we found that type 2 diabetes does not affect cognitive decline between visits when compared to Alzheimer's with no presence of diabetes. We found no significant differences overall in the uniform dataset test scores analyzed in the present study. Thus, our central hypothesis was rejected, and the alternative hypothesis that type 2 diabetes doesn't affect cognitive decline in Alzheimer's patients seems the most likely. Due to the large patient population and significant health effects that both diseases have, there have been an extensive number of studies done that have yielded mixed results on the relationship between the two. There are still many unknowns about the underlying pathophysiology of Alzheimer's disease alone, as well as was shown in the 2018 meta-analysis that found vascular mechanisms might possibly play a larger role in cognitive decline than the AD-specific pathology that we all know. Moving forward, I would like to note the present study focused solely on the presence of type 2 diabetes as a factor that could affect cognitive decline in Alzheimer's disease. Other demographic variables, such as ApoE alleles, especially the ApoE4 allele, which is associated with an increased risk of Alzheimer's disease, or whether patients were other diabetic medications were not used to group the subjects. Thus, future study designs could incorporate these as well as additional variables to possibly identify differences in cognitive decline progression in Alzheimer's disease patients. Finally, here are my references listed that I used throughout my project, and thank you very much for listening to my presentation.